Dr. Shane Hegarty is the Chief Scientific Officer and founder and IP inventor at Boston-based biotech AXONIS Therapeutics. This highly productive researcher has over 30 research papers and over 1,200 citations, as well as over 13 years of experience in cutting-edge, award-winning neuroprotective research, neuromodulation, and neuroregeneration research for neurological disorders. We're pleased to have him here today with his teaching experience as a faculty member in the neuroscience department at the University College Cork in Ireland. And it's my distinct pleasure to introduce Dr. Shane Hegarty.
Thank you very much for the kind introduction. Just checking everyone can hear me and they can see my slides.
Yeah.
Perfect. And also just want to express my appreciation for getting the opportunity to join this roundtable today. It's a privilege and an honor. And I'm sorry I couldn't be there in person, you know, with the great panel and speakers and group that you brought together. Clearly in an absolutely fantastic setting as well for the meeting. So I hope I can contribute today to pushing new therapeutics forward for SYNGAP1 haploinsufficiency disorder. So today my talk is quite focused in a way. It's really talking about therapeutically targeting KCC2 to restore excitation, inhibition, imbalance pathologies in neurological disorders, which has relevance for many different neurological disorders, and I hope to explain that today well. Just a quick overview of AXONIS. We're Boston-based, and we focus on first-in-class small molecule CNS therapeutics. Our lead program are these KC2 potentiator drugs, and we have a second program also working on neuroprotection. So our team is spearheaded by Joanna Stanica, who is our CEO and co-founder. Joanna and I were research fellows in Harvard Medical School and Boston Children's Hospital in the laboratory of Professor Zhigang He, from which many of these discoveries in that lab have been spun out through AXONIS, and I'll talk a bit about that today. Our executive chairman and co-founder and chair of our scientific advisory board is Dr. Corey Goodman, who is a former neuroscience professor and very esteemed in that field, but in more recent years has become a serial drug developer and managing partner at VenBio. I head the neuro discovery team, and we also have a number of consultants to inform us on our drug development effort. So the idea behind AXONIS was to develop medicines to treat fundamental pathologies, and we focus on three pathologies we believe are shared across many neurological disorders. The first is excitation, inhibition, imbalance in neuronal circuits. The second is the degeneration of neurons. And the final pathology we were interested in was the loss of neuron connections and why they don't self-repair. And what we worked on was therapeutic paths to manage these pathologies. So restoration of EI balance, neuroprotection, and axonal regeneration. And the way we went about discovering new therapeutic targets for these pathologies was from in vivo phenotypic screens. The reasons we performed our screens in vivo are because the central nervous system is very complex. These neurons are in complex wiring systems. There are different regions of the CNS and spinal cord that have different functions. There are many different multi-functioning cell types and also if we think about different organ systems interacting with one another, for example, the immune system and the brain. And our screening process took two different approaches. One was using AAV CRISPR screening and a forward genetic screening approach, which I won't talk too much about today, and others involved using tool compounds to perform screens as well. And based on the hits, the most potent phenotypes, we then pursued drug development efforts through AXONIS. So these screens were performed academically in the laboratory of Professor Zhigang He in Boston Children's Hospital and Harvard Medical School. Over 12 years ago now, Zhigang performed a seminal screen where he asked the question whether neurons could repair themselves through genetic manipulation. Before that, the consensus in the field was that axons don't regrow because of the external environment, And he disproved that dogma by showing you can manipulate particular genes, in this case P10, to cause a cell-autonomous axonal regeneration. But he didn't stop there, and later there were two other screens which I was part of and leading. The second was the small molecule screen in spinal cord injury model, which I'll talk a bit about today, where we discovered KC2 as a therapeutic target, and more recently a genome-wide AAV CRISPR screen to identify novel targets that can prevent neuronal degeneration, which is the other programs in the company. So just quickly to go over our in vivo screening approach using AAV CRISPR platform, the main bottleneck to doing these screens is actually the generation of the AAVs and the CRISPR library themselves. Obviously, we've completed that now, and the CRISPR plasmids can be repackaged into different AAV serotypes to target different cell types and regions of the brain, depending on the route of administration and promoters and things like that you can also modify. But the beauty of this is the cell type and selective effects you can achieve sometimes with AAVs, depending on your model and cell type of interest. And then when you combine that with neurodegeneration or other neurological disorder modeling, you can unbiasedly assess what genes can provide therapeutic benefit when you target them. And CRISPR can be used to delete genes, it can be used to activate genes, and there's different tools here to really examine unbiasedly how to discover new targets for neurological disorders, where you focus on the downstream pathologies of a disorder. So what I want to focus on today really is, how do we come across KC2 in the first place? And that was a breakthrough discovery in the spinal cord injury model, again another in vivo screen. In this case, we wanted to model the clinical reality where spinal cord injury patients have a lot of spare tissue in their spinal cord, which traverses the injury site, but that tissue is non-functional, it's dormant. And we asked the question why that happens, and it can be reactivated again clinically with epidural electrical stimulating devices. So we want to examine the molecular basis for this dormancy and whether we can reactivate this relayed tissue across the spinal cord to try to restore benefit in this pathology. The model was labor intensive, but we combined that with systemic administration of small molecule drugs that have a couple of properties. First, they needed to be able to cross the blood-brain barrier to assess their therapeutic effects. And we also focused on small molecules that target neuromodulatory proteins. So the idea being, can we reactivate this dormant spare tissue that has been demonstrated with electrical stimulation? So we focused on receptors, channels, transporters, whatever targets that we have at least two compounds for, or even drugs on the market. And then we did an unbiased screen. But just to be wary, because the model was very labor intensive, we couldn't screen as many things as we'd like. But we use also a severe model to ensure that the baseline didn't have any spontaneous recovery, which we can see here. Because of the fact that there's no brain-derived innervations to the lower spinal cord because of the double hemisection, the control mice don't regain any spontaneous stepping ability. However, when we either initially used the tool compound CLP290, that was the only compound that was able to reactivate the spare tissue to cause stepping ability recovery in this model. We followed that up with multiple genetic approaches and AAV approaches to show that this benefit in this model was based on increasing KC2 function. So through pharmacological and genetic approaches, we demonstrated that we can reactivate that dormant spare tissue, in this case, inhibitory interneurons in the spinal cord, to lead to motor function recovery. And that was our expertise, that was our passion project at the time in spinal cord injury, but KC2 goes beyond spinal cord injury as a therapeutic target. It is the chief CNS-specific chloride transporter that is essential for GABA mediated inhibition of neurons. As we can see here, if we compare KC2 to other family members in this transporter class, we can see it's the one that's selectively expressed in the CNS, which is a very attractive therapeutic target profile for a CNS disorder. And if we can look here on the other side of the screen, where we can see that there has been many drugs developed for targeting the GABA inhibitory mechanism. However, we want to develop the first-in-class therapeutic for KCC2. We believe it can unlock the full potential of the GABA pathway in terms of therapeutic modalities. And we also aim to leverage the successful history of drug development and translation on this fast synaptic inhibitory pathway. And we'll get into that a little bit later as well. So what happens when KCC2 function is impaired? And it occurs in a conserved mechanism in a couple of different disorders. So downstream of excitotoxicity or inflammation, what has been demonstrated to occur is KCC2 reduced expression. And when KCC2 is not on the membrane pumping chloride out of neurons, the GABA receptors don't function the way they should. So GABA receptors are ion channels. They do not dictate the direction at which chloride flows. So if KCC2 isn't functioning appropriately, GABA is no longer inhibitory and can even become excitatory. So you can see how that could lead to excitation, inhibition, imbalances in different disorders. And these conserved mechanisms of excitotoxicity and microglia occur in different disorders. There's also other neurodevelopmental disorders where we see, because of delayed brain maturation, you do not get an increase in KCC2 expression as you should, and similar mechanisms of EI imbalance can occur where GABA doesn't inhibit as it should. So this is why KCC2 has a key role in development and can be implicated in neurodevelopmental disorders. KCC2 is the master regulator of the switch of GABA from being excitatory to inhibitory during development. This happens in the postnatal period where KCC2 expression increases over time. And due to KCC2 expression increasing and the chloride concentration lowering within neurons, GABA stops being excitatory, which it is in early development, and switches to become inhibitory, which is the more accepted role we're all familiar with. So you can see the essential critical role KCC2 has for appropriate inhibition in the central nervous system. And that can be studied in genetic models of KCC2 hyperfunction. So knockout mice, if you have mice with knockout KCC2, they're, first of all, they don't survive very long. They have breathing difficulties, they have seizures. You can make hypofunction mutants or more specific mutants of KCC2. Again, common problems are seizures, behavioral issues, anxiety, breathing issues, and that's been well studied. And on top of this, there's been reports of KCC2 downregulation or mutation causing neurological disorders in humans. That's been looked at in ex vivo brain tissues in the case of neurotraumas. It also has been observed in neurodevelopmental disorder tissues and CSF in the case of Rett syndrome, and also some resected tissue or postmortem tissue from different epileptic disorders. And critically, and this has been demonstrated now for over a dozen patients, loss of function KCC2 mutations themselves cause severe epilepsies, refractory seizures, and they cause autism. Well, as we all accept, they cause intellectual disability that typically gets diagnosed as autism. And there's been a number of mutations in KCC2 associated with psychiatric disorders. So this is becoming more and more well studied over the last decade. And there's been now over 700 KCC2 mutations identified. They haven't all been as prescribed or assigned as pathological, but further work is needed there. And what we can do now is we can back translate some of these mutants and study them to show why do these mutations cause seizures and intellectual disability and other problems. We can also use the models and perform forward translations to understand what happens when there's KCC2 hyperfunction. Well, usually what happens is there's impaired chloride extrusion in the neuron, and the neurons no longer respond to GABA the way they should. And that can be studied in vitro, it can be studied in tissue slices, it can be studied in animals, and it's been widely studied in multiple different models and tissues and neurological disorders associated with the CNS. And really what this tells us is that KCC2 is a genetically validated target, and that human pathologies are recapitulated in models when we back translate from the clinic. And just because of its broad applicability in excitation inhibition imbalance disorders, it has the potential to treat many disorders. We're not listing all of our plans here for confidentiality reasons, but we can get an idea of the impact that KC2 potentiating therapies can have in different pathological situations. We obviously historically did a lot of work in spinal cord injury and neuropathic pain as well. And all you need to do really is to go on to PubMed and search KC2, and you can find multiple different groups, multiple different neurological disorder models, and even, as I said, some clinical papers looking at the patients with KC2 mutations to see how important KC2 is for a proper CNS function and how it plays a role in the pathology of EI imbalance disorders such as spinal cord injury, neuropathic pain. We can study this electrophysiology using electrophysiology such as EEG, other spinal reflex biomarkers as well. Their developmental disorders are starting to be well studied, multiple different epilepsy models, as well as neurotrauma models. And people use different tools here. They can either show that KC2 is downregulated in these disorders, contributing to the pathology, or they can use genetic or tool compounds to try to rescue KC2 function to provide benefit in these models, depending on what the pathology is. We do that here in our own lab, which we haven't shared widely yet, and it's been published by multiple other groups. So it's becoming well accepted and well conserved that KC2 is a validated target for providing benefit in models of neurological disorders. So what are AXONIS doing now? We want to have a discovery engine, or we've built a discovery engine to fully unlock the therapeutic potential of this space. So that involves having KC2 drug screening assays, binding assays, mechanism of action assays. It is a difficult study to target. It's a transporter, but we've developed tools to be able to do this effectively. We're looking at different biomarkers, hopefully translational biomarkers that we can also use in the clinic to validate on-target activity of our KC2 potentiators, and hopefully they're also tied to pathology to give an early sign of clinical benefit. We're perform multiple in vivo and in vitro assays looking at KC2 potentiation, and we've recruited many of the experts in this space. So Yves is one of the first people to discover how important KC2 was in neurological disorders, and Clifford Wolf also is in that category. And Jigang was performing the screens we spoke about a bit earlier. I was working in his lab with Joanna, where we first saw that in spinal cord injury, at least, it provides the most functional recovery than any other approach we've ever tried. It beats anything we've tried before. So we're excited about that space as well. So in case of spinal cord injury, our oral KC2 potentiator drug could have multimodal therapeutic benefits because of the fact that the excitation inhibition imbalance is causing multiple pathologies at once, depending on what tissue is being affected. So we've demonstrated in the case of the local inhibitory neurons traversing the injury site that their disinhibition and their inability to transmit brain-derived signals to the lumbar spinal cord is leading to paralysis. And KC2 restoration, KC2 function can reactivate that tissue and relay motor signals to provide stepping ability in the animals. In the case of neuropathic pain, it's been widely studied before, and we've reproduced these results in our own lab, that KC2 provides analgesic benefit to similar levels as pregabalin, which is a first-in-class medication for neuropathic pain. And a key thing about these treatments is they don't cause sedation. Because they're not causing a global repression of neuronal activity, they're basically fine-tuning the neurons to respond to the endogenous GABA. So it's a physiological inhibition is restored. And in spinal cord injury, there's also problems on the ventral motor neurons because of their EI imbalance, which leads to spasticity. So spinal cord injury patients, 2/3 of them will have comorbidity of neuropathic pain and spasticity because there is EI imbalance in the damaged spinal cord, which we try to address with our KC2 potentiator drugs. In the case of epilepsies, and this is much more studied than spinal cord injury as a mechanism, multiple authors, multiple groups working on this mechanism, and it seems to be conserved across multiple different epilepsies. So what leads to KC2 hyperfunction in the first place? As I mentioned earlier, it can be a genetic EI imbalance. So, a neurodevelopmental disorder where brain maturation isn't occurring as it should, or you can have a local excitotoxicity or neuroinflammation that can be from a traumatic injury, for example, which is one of the causes of medial temporal lobe epilepsy. And this leads to KC2 hyperfunction, which leads to a lack of appropriate neuronal inhibition that contributes to seizures. What's also been shown is that seizures themselves, status epilepticus, and NMDA receptor activity, this is actually driving a cycle of further KC2 hyperfunction, which exacerbates the severity of the seizures, which makes them pharmacoresistant to the drugs on the market, especially those drugs that would target GABA-mediated inhibition. So KC2 is a novel but well-studied epilepsy target for addressing drug-resistant seizures, and we're excited about that direction as well. In the case of SYNGAP1 haploinsufficiency, last year there was a paper where we saw one of the first reports for a potential role for KC2 hyperfunction in this neurodevelopmental disorder, where when KC2 expression is supposed to be increasing in the postnatal period to allow appropriate GABA inhibition in the brain, in the mouse model anyway, we can see a hypofunction or loss of KC2 expression when it should be there. One of our principal investigators, or our only principal investigator, correct on that one, Dr. Shilpa Gadam is an expert in this space. She's published on this before. Obviously, in this case, not on a KC2 related mechanism, but we have the ability to study this well. We've actually been awarded a grant from the SYNGAP Research Fund to collaborate with Dr. Jin Tang in Boston Children's Hospital and AXONIS Therapeutics will collaborate on a project to look at the effects of KC2 enhancing small molecules, including nerve compounds, but Jin Tang himself has done this work before in Rett syndrome. So he's got the tools to perform it. He's got the assays where he can study KC2 hyperfunction in patient-derived iPSC neurons. And then we can look at whether our compounds, if there is a pathology, we can study whether our compounds can rescue that. So that project has just got off the ground. But, you know, if we look at this and we look at other neurological disorders, we can, you know, at this point, propose that KC2 may be a promising therapeutic target for treating EI balance that occurs in SYNGAP1 deficiency disorder. So all that really just boils down to what we're trying to develop as a company, and that is the first oral brain penetrant KC2 potentiator drug into the clinic. And we're working tirelessly to make that a reality. And we believe that these compounds could really unlock the full potential of the existing GABA drugs on the market and can be a blockbuster drug in itself. We talked about the widely applicability of the pathology that can occur if KC2 function isn't at the normal levels. And we imagine the benefit in two ways. So when we use it as a monotherapy, which we most commonly do, we believe we are rescuing neurons' ability to respond to the endogenous GABA. So we're restoring balance to that tissue. But you can see scenarios where when you combine KC2 with GABA-A targeting compounds, and this has been published before, that you can increase the efficacy of these drugs or restore the responses to these drugs. So we can think of many drug-resistant epilepsies where benzodiazepines or phenobarbital or things like that, they don't work. And they may not work because the GABA receptor can't respond to the drug in the first place because KC2 hypofunction. So that's a powerful adjunct therapeutic in that setting. And in the same way, we can increase efficacy of the drug and reduce the doses of these sedation-causing compounds as well, which so we can reduce side effects. So we're excited about those directions, and we're currently proving those out. I'd just like to acknowledge the network it takes to make this project. We have many collaborators, advisors. We've raised over $6 million in non-dilutive grant funds to work on these new therapeutics, and without their support, we wouldn't have made this progress so far. And we are about to translate cutting-edge neuroscience into impactful medicines, and I'm excited to take any questions if there are any.
research. I'm just curious if there's any considerations for modulating KC2 via upstream regulators like PDNF.
That is certainly a mechanism that would work, but you'd have to then consider the off-target effects of working on the TrkB receptors or working on these other kinases like Wnt. They have other effects in the cell. So ideally, you want to target KC2 directly, which is what we're doing. And but these other mechanisms, they may be needed in different pathologies. So certainly other modalities, other classes of compounds could use such mechanisms to achieve therapeutic benefit. And that's been published before, that those approaches work.
Hey, that's great talk. So I have 3 questions. So the first one is related to the ability of CLP219 that you are sort of envisioning as a CCC2 activator. So, achieving cell type specificity with these wide range drugs is very difficult. So AXONIS in the future, does it envision to sort of incorporate or selectively improve this function in a cell type specific fashion? Now we know that CDF1 is not only present in excitatory neurons, but it's also present in GABAergic neurons. And there is increasing evidence of that. So that's the first question. Second question is, so one aspect of modulating GABAergic signaling is by regulating the amount of NKCC1 as well. So, and in several developmental disorders, NKCC1 has been considered to be a druggable target. So, is there sort of a future direction that, whether instead of looking only at KCC2, whether NKCC1 can also be selectively modulated? And the third question is basically about, actually, it's okay. If you can shed light on these 2 questions, then thank you so much.
Fantastic questions. And, we'd love to, work at a cell type specific level, but right now with our small molecule compounds, they get into the brain and they don't obviously target particular cell types. So that's the reality of the small molecule drug, but it's orally bioavailable and it gets sufficiently into the brain. So on the cell type specific part, this is how I think about these pathologies. And, I know there's stochastic natures of, different cell types having different levels of a pathological protein. So therefore, in the case of KC2, different cell types will have more KC2 hypofunction than others. So the mechanism of action is to rescue the function of KC2 in the cell types who have the hypofunction. So that's how we can treat the deficits in the cells that have the deficit. And in the normal cells, there's a lot of regulatory mechanisms to avoid, KC2, there's a saturation effect basically in the healthy cells. So that's how we can get at, targeting the cell types that need a KC2 potentiation treatment. On the NKCC1, another exciting target. So the primary issue on that target is it's expressed in many different tissues. So it makes it less attractive therapeutic target, but it's another means to do the same thing in terms of restoring chloride homeostasis. So for example, and I've studied this as well, I used to be very close to the infant center in University College Cork who did a clinical trial on bumetanide. And the problems were the children, because NKCC1 has an important role in the epithelial cells of the ear, they were having hearing problems. So ideally, if you're working on chloride homeostasis, you want to work on the target that's selectively expressed in the CNS, and that's KCC2. So you could have therapeutic benefit with the NKCC1 approach. However, you would have to deal with the side effects. Also, bumetanide is very poor at getting into the brain, which means you need to use higher doses. So again, you're ramping up the side effects even more there. So it's not a bad approach theoretically, but I don't see why it would be better than a KCC2 potentiation therapeutic. So I hope that answered your questions.
Dr. Hegarty, thank you for your time. As a spinal cord injury patient myself, it's exciting, but also from the perspective of the neurological disorder community and SIDGAP1 opportunities as well. Thank you for your work.
Thanks, Matt.